like, subscribe and hit the bell icon for prior notifications.
Hello everyone, today I am going to discuss about different biochemical events which occur during the process of fertilization. So far we know that fertilization is the fusion of two haploid gametes for the production of a diploid zygote, and there are certain pre-fertilization events which occur before fertilization such as insemination and capacitation. In my previous video I have already discussed about insemination and capacitation in detail. So after capacitation, about 200 sperms, only 200 sperms. During insemination, millions of sperms are deposited inside the vagina of the female, but among those millions, only few hundreds of sperms are selected by the process of capacitation, and they are able to move through the female reproductive tract and able to reach the surface of secondary oocyte for fertilization. Suppose you think this is the secondary oocyte. I have already drawn a part of the secondary oocyte which is waiting inside the ampulla of the fallopian tube. We know that after ovulation, the secondary oocyte is released into the peritoneal cavity, and from the peritoneal cavity, fimbriae takes up the secondary oocyte into it and it reaches the ampulla, and the growth or division of the secondary oocyte is arrested at metaphase 2 of meiosis II. So now the secondary oocyte, the arrested secondary oocyte is waiting inside the ampulla, and many sperms, they have reached the surface of the secondary oocyte to fertilize it, and these are the outer boundaries or outer membranes of the secondary oocyte through which the sperms has to reach the surface of the plasma membrane of the oocyte. But remember, only one sperm enters the egg. Only one sperm finds its way to reach the surface of the oolemma or plasma membrane. Why one sperm? Because if more than one sperm enters the egg and there is fusion of more than two nuclei other than these two, this is the egg nucleus and this is the sperm nucleus. So these two are combined to form 2N or the nucleus of the zygote. If more than these two nuclei they are fused, then this results in chromosomal abnormality. That means certain polyploid conditions arise and an abnormal zygote will be formed at the end of fertilization that is not able to form an embryo. So that's why only one sperm enters the egg. Several sperms, they have now reached. Suppose you think this is the first sperm to establish contact with the corona radiata layer. So this is the corona radiata layer. These are the corona radiata cells which are held together by means of a ground substance that is called hyaluronic acid. And this is the zona pellucida layer. Inner to the zona pellucida, this is the perivitelline space. And this is the outer boundary of the oocyte, that is oolemma or vitelline membrane. And these are the cortical granules, and this is the oocyte nucleus. So what will happen now? This is the first sperm which has reached the surface of the suppose these are the corona radiata cells. Before this stage, suppose this is the beginning. It first reached or touched the corona radiata cell layer. Then what will happen? It will try to penetrate. So that is called penetration by the sperm. So penetration is a continuous process till it enters the egg completely. So first is the penetration by the sperm, which is the result of acrosome reaction. Remember, acrosome reaction occurs before penetration, which enables the sperm to enter the membranes of the egg. So what is acrosome reaction? Acrosome reaction, that means the reactions which occur over the acrosome, that is called, or those are called acrosome reaction. The main event of acrosome reaction, or the main thing what happens during the process of acrosome reaction, is a release of different enzymes. A release of different enzymes from the acrosome. They are together called sperm lysins. The enzymes the acrosome contain are called sperm lysins. So we know that acrosome is the modified Golgi body which forms the anterior part of the sperm head, and it is also called megalysosome. Megalysosome. So it contains obviously lytic enzymes, and those lytic enzymes are called sperm lysins. And there are mainly 3 enzymes. Number 1, it is hyaluronidase. So this enzyme digests the hyaluronic acid present between the corona radiata cells, so that the bond between the corona radiata cells will break. And next is corona penetrating enzyme which digests the corona radiata cells. Then final enzyme which is called acrosome. Acrosome, or it is the jonalysine. Jonalysine, which digests the zona pellucida layer. Suppose the sperm has entered through this corona radiata layer and has touched the surface of the zona pellucida. Here there are certain specific receptors called JP3 receptors. Zona pellucida type 3 receptors, with which the sperm head binds. And there are certain structural modifications occur over the sperm head as well as the zona pellucida. And finally, the acrosomal contents are released and the zona pellucida is digested, and the sperm reach the surface of the vitelline membrane. So this is the simultaneous event, one after another it is occurring. First, the sperm establishes contact with the corona radiata layer. Then, after digesting the hyaluronic acid and corona radiata cells, the sperm now reaches the surface of zona pellucida. Then it binds to JP3 receptor. Then acrosine or jonalysine, it digests the zona pellucida layer. Then the sperm reaches the surface of the vitelline membrane. So, and remember, this acrosome reaction occurs under the influence of certain ions like magnesium and calcium. Calcium ion is highly essential, and proper pH and proper temperature is also required. So now the sperm has reached the surface of the vitelline membrane or oolemma. Now what will happen? Here there will be formation of a reception cone. This is the first event of activation of oocyte. So now when the sperm reaches the surface of the vitelline membrane, the oocyte is activated, and the first thing which is done over the membrane is formation of reception cone. So this is a small projection which occurs over the vitelline membrane to receive the sperm is called cone of reception. Cone of reception formation. So this is a part of activation of oocyte. So now there is fusion of the membranes, the outer membranes of sperm with the plasma membrane or oolemma here. And by phagocytosis method, what will happen? All part of the sperm enters the egg. Remember, in all mammals along with the humans, the tail, almost all part of the sperm enters the egg. So through the cone of reception, the sperm enters the egg. So now what will happen? Immediately after entering the sperm, after entrance of the sperm, there will be a structural modification, or there will be a modification over this membrane to block the entrance of further sperms. So there is block to polyspermy. Polyspermy is entrance of more than one sperm. So there is block to polyspermy soon after entrance of the first sperm. So why there is block to polyspermy? I have already discussed that if more than one sperm enters, then chromosome number of the zygote will be disturbed. So there is block to polyspermy. So there are two mechanisms. One mechanism acts suddenly, preventing more sperms to penetrate, that is called fast block to polyspermy. And after fast block mechanism, another mechanism acts, that is called slow block to polyspermy. So what is polyspermy? Understood. Polyspermy is entrance of more than one sperm, which will disturb the zygote, the chromosome number of the zygote, resulting in polyploid condition. So there will be blockage system, blockage mechanism. One mechanism is there which acts suddenly after entrance of the first sperm, that is fast block mechanism, and after the fast block mechanism there is slow block mechanism. And another thing happens, there is completion of meiosis 2 completed, and ovum is formed, mature ovum is formed, and second polar body is released into the perivitaline space. So what will happen under fast block, under slow block? So what is fast block? Remember, when the sperm enters through the plasma membrane of the oocyte, the membrane potential is minus 70 millivolt. This is the resting membrane potential. The sperm enters, and when the sperm fuses, the membrane of the sperm fuses with the oolemma and it enters, it gradually increases, the membrane potential gradually increases and it reaches up to plus 30 millivolt. That means the oolemma is now depolarized. After the entry of one sperm, the oolemma is depolarized, and at this changing potential, further sperms cannot penetrate. The sperms can penetrate the egg only at this potential, this membrane potential. So when it reaches plus 30 millivolt, around plus 30 millivolt, then the sperms cannot enter. So this is fast block. Soon after the entrance, this mechanism acts, blocking polyspermy. Then slow block. What will happen during slow block is, these are the cortical granules. So from endoplasmic reticulum, these are endoplasmic reticulum, calcium stores are released. Calcium ions are released in large amount, and they stimulate the cortical granules to fuse with the plasma membrane of the oocyte. So here you can see the cortical granules have been fused with the plasma membrane, and they release their content into the perivitaline space. So these contents, what they do? There are certain enzymes here. They repair these entrance points, making it more rigid so that more sperms cannot enter. That hard rigid membrane which is formed at the point of entrance over the vitaline membrane is called fertilization membrane. So there is fertilization membrane formation. So what is fertilization membrane? It is the area it is the rigid area present over the vitaline membrane which is formed just after the entrance of the first sperm, and it is the repaired part by the contents of the cortical granules. And another thing also happens, these granules break the bond between the zona pellucida. Here they break the bond between the zona pellucida and the vitelline membrane, so that the zona pellucida and vitelline membrane are detached from each other, and more than other sperms they cannot enter the oocyte. So that is called slow block. So let us erase all these things and summarize what we have discussed. So there is slow block to polyspermy and fast block to polyspermy. Fast block. Under fast block there is depolarization of oolemma. Slow block, there is cortical reaction which occurs over the cortical granules, during which what happens, there is fertilization membrane formation. Fertilization membrane is a rigid membrane, fertilization, which is formed on the entrance point over the vitelline membrane. Fertilization membrane formation and the zona pellucida is detached from oolemma. So these things happen. So after that what happens inside the oocyte? So soon after entrance, the outer membrane of the sperm is disintegrated and all the contents are released. All the sperm contents are released inside the ovum. So now it has already turned to ovum. Meiosis 2 has been completed. So tail is destroyed. The mitochondria, the other part, all other organelles are destroyed except the nucleus and the proximal centriole. Remember, all the organelles of the sperm are destroyed inside the ooplasm except the nucleus and the proximal centriole. So now the male nucleus gets nourishment from the ooplasm and it is slightly enlarged. That is called male pronucleus. Simultaneously here the oocyte nucleus also is enlarged and it is called the female pronucleus. And there is fusion of, let me draw here just. So suppose this is the proximal centriole only and this is the large male pronucleus and this is the large female pronucleus. This contain haploid chromosome and this also contain haploid chromosomes. So they move towards each other. The two pronuclei, they move towards each other and finally their plasma membrane fuse. And the chromosomes, suppose here there are chromosomes and here there are chromosomes. So they are fused and the nucleus become now 2M. And this structure is called zygote. So this is all about fertilization, which results in the formation of zygote. This process of mixing of these chromosomes is called amphimixis or karyogamy. Amphi means both nuclei they fuse, that's why it is amphimixis or it is karyogamy. Karyon means nucleus, gamos means marriage. So there is marriage of two nuclei, you can say. So this is karyogamy or amphimixis. So during fertilization mainly three things occur, three events, three gamete occur. That is the syngamy here. Syngamy is the fusion of the plasma membrane and there is plasmogamy. Plasmogamy means there is a fusion of the cytoplasm. the little amount of cytoplasm present in the sperm that is fused or mixed with the ooplasm, that is plasmogamy, and last is the amphimixis or karyogamy. So 3 gametes occur during the process of fertilization: syngamy, plasmogamy and karyogamy.